The quality of literature plays a significant role in analysing the application of TDCSs on autism spectrum disorder. The quality of literature plays a significant role in analysing the application of TDCSs on autism. As stated by Woods A.J. 2016 et al., TDCs are described as a non-invasive brain stimulation techniques increasingly used for modulation of central nervous system excitability in humans. Additionally, a relevant study by Kang J. et al. 2016 stated, ASD is a brain disorder which TDCSs can be used to improve and modulate motor and cognitive function in an individual, as well as another study by Osori AAC and Brunini AR 2019 arguing TDCSs can also be used to improve symptom severity in ASD. We can explore the quality of literature through discussing the physiological mechanisms of ASD and the physiological effects of TDCSs and how they propose to influence physiological effects of ASD. So what is ASD? We can further explore this through discussing the physiological mechanisms of ASD. The topic of ASD is not well understood, however there are possible theories to further describe the mechanisms involved. A 2020 study highlighted the importance of how one obvious challenge in understanding how ASD develops is the wide range of mechanisms associated with it. In addition to this, a 2006 article outlined the point that for audiences to understand the mechanisms of ASD, they must split its development into two sections, the pathophysiology of brain structures and its mechanisms linked to autism, and the neurophysiological connections between the brain structures and behaviours. As stated by a 2008 article, it believes ASD, in comparison to many other brain disorders, autism lacks any unifying clear pathology at the molecular, cellular, and systems level, and goes on to further discuss the complexity and broadness of the ASD phenotype. A synaptic study analysed ASD and its mechanisms, stating genes that affect synaptic maturation are implicated, resulting in neurobiological theories focusing on connectivity and neuroeffects of gene expression, which further establishes the connection between neurochemicals and autism not being well known. A study outlined that fMRI brain scans have shown abnormalities in amygdala, hippocampus, septum, malia bodies, and the cerebellum of the brain, which can further show that autistic symptoms may be a consequence of abnormal function in these structures. The study also elaborated how there's an increase in a major neurotransmitter, serotonin, which affects potentiation at synapses and may play a role in the development of nervous system. This establishes the standard of the literature plays in a role in explaining the application of TDCSs on ASD. So what are the physiological effects of TDCSs and how are TDCSs proposed to influence physiological mechanisms of ASD? Numerous studies show that there are many positive effects of TDCSs on ASD. A study argued that having a solution such as TDCSs for ASD could lead to increased attention, productivity, and quality of life, and how TDCSs offer reliable physiological internal state monitoring for ASD individuals. A systematic review investigated the use of TDCSs in ASD through identifying peer-reviewed scientific studies concluding that outcomes addressed ASD symptoms and neurophysiological functions, and meta-analysis synthesis identified improvements in social, health, and behavioural problem domains in the autism treatment evaluation checklist. Additionally, a review conducted its own randomized controlled trial, which involved 105 young adults with ASD being showed multiple sessions of TDCS's prototype. The study's outcome presented TDCSs were effective in improving social functioning in adolescents and young adults with ASD. A 2020 pilot study also showed that TDCS is to be an effective therapeutic approach towards ASD in terms of improving sociability, behaviour, health, and physical conditions with no reported side effects. In contrast to the following studies, there are studies that argue the negative physiological effects of TDSs. A current meta-analysis evaluated the effectiveness of TDCSs in individuals with ASD, which found most studies to have moderate to high levels of bias and a lack of ongoing of treatment effects. It urges caution is needed due to the quality of original studies and publication bias, and recommends large trials and follow-up observations are required to evaluate TDCS's effectiveness in ASD. Another similar article found TDCS's evidence is sparse and of low quality and compels for future randomized controlled trials in order to draw conclusions regarding TDCS's efficiency in pediatric samples with ASD. From a different perspective, a recent study argued that studies shouldn't focus on the cause, eliminating or reducing symptoms of ASD, as it makes people with ASD feel like they've got a problem when they should feel accepted with the condition. It also argued that the only reason studies want to find out the cause or the cure of autism is to eliminate it, either through a cure or aborting fetuses identified to have a high risk chance of being autistic. We keep sinking research money into the why of autism, which further worsens mental health outcomes for autistic people. This overall proves the quality of literature plays a crucial role in explaining the application of TDCSs on ASD. The standard of literature plays a compelling position in discussing the application of TDCSs on ASD. For exploring the standard of literature presented to us, we are adequate in understanding how there is quality evidence that supports the application of TDCSs on ASD to a certain degree. However, its emphasis on reducing symptoms or finding the cause of ASD through the use of techniques such as TDCSs may be damaging to the mental health of individuals with ASD. It is through these notions that we are capable of understanding the significant role literature plays in discussing the application of TDCSs on ASD.